Hello and welcome to the Talking Techniques podcast, brought to you by Biotechniques. This show brings you the latest from the frontiers of the life sciences, straight from the people exploring them. I'm your host, Biotechniques digital editor Annie Colson, and in this episode, supported by Cynobiological, we'll be discussing cytokines, their role in the immune system and how they can be harnessed for therapeutic and diagnostic purposes in immunotherapy. Coming up on the podcast, we learn about cytokine dysregulation in disease.
Any kind of dysregulation or imbalance within cytokine networks can have profound implications for immune homeostasis and disease pathogenesis.
How this knowledge can help improve patient outcomes.
So, understanding the cytokine biology and its impact on immunotherapy outcomes helps in the identification of biomarkers for predicting the treatment responses, also guiding treatment selection and personalizing therapeutic approaches.
And what the future holds for the role of cytokine diagnostics in immunotherapy.
So I think cytokine diagnostics have the potential to revolutionize precision medicine in immunotherapy.
Our guest today is Ritvika Biswas, a field application scientist at Cynobiological. Ritvika, it's great to have you on the show.
Hi, Annie. Thank you. It's great to be here.
So to start us off, could you tell us what cytokines are and their role in the body?
Yes, sure. So cytokines are small proteins secreted by various cells in the body, mainly immune cells. And these proteins play a crucial role in cell signaling and communication. These signaling molecules are involved in regulating immune responses, inflammation, and other physiological processes. In the body, cytokines act as messengers that coordinate interactions between different cells of the immune system, as well as between immune cells and other cell types. They can stimulate or inhibit the activity of immune cells or promote cell proliferation or differentiation and regulate the production of other cytokines. Overall, cytokines serve as key mediators of the immune system, contributing to body's ability to mount effective immune responses against pathogens, maintain immune homeostasis, and resolve inflammation. Their dysregulation or imbalance can lead to various diseases, including autoimmune disorders, inflammatory conditions, and even cancer.
And more specifically, what is their role in immune regulation and signaling?
So cytokines play a central role in immune regulation and signaling, mainly by mediating communication between different cells of the immune system. So here are some key aspects of their roles, like, for example, cellular communication. So cytokines serve as signaling molecules that allow immune cells to communicate with each other. And they are produced by one cell and bind to receptors on the target cells, triggering a specific cellular response. Cytokines can also activate immune cells, such as T cells, B cells, macrophages, as well as dendritic cells, by promoting their proliferation, differentiation, as well as functional activation. For example, IL-2, that is a cytokine that stimulates the proliferation and activation of T cells. Cytokines can also play a crucial role in initiating and regulating inflammation, which is the body's response to infection. And pro-inflammatory cytokines such as the TNF-alpha, tumor necrosis factor, and interleukin-1 promote inflammation, while anti-inflammatory cytokines such as the interleukin-10 help to resolve the inflammation and maintain immune homeostasis. Cytokines can also regulate various aspects of immune responses, including the generation of immune memory, the recruitment of immune cells to sites of infection or injury, and the elimination of pathogens or infected cells even. They also play a role in immune tolerance, preventing the immune system from attacking the body's own tissue, because that will be a huge problem, right? And then cytokines can also help to maintain a balance between different subsets of T helper cells, like the T helper cell 1 and T helper cell 2 cells, which basically produce distinct cytokine profiles and are involved in different types of immune responses. So imbalances in Th1 and Th2 cytokine production can contribute to autoimmune diseases, allergies, and other immune-related disorders. So overall, cytokines serve as critical mediators of immune regulation and signaling, basically orchestrating the complex interactions between different cells of the immune system and contributing to the body's ability to mount effective immune responses against pathogens while maintaining immune homeostasis.
Can you discuss the complex interactions between cytokines and cytokine networks and how they collectively orchestrate immune responses?
Absolutely. So the interplay between cytokines and the cytokine networks is a very sophisticated communication system within the immune system, where various cytokines act as messengers to regulate and coordinate the immune responses. So think of cytokine networks as intricate webs of interactions where different cytokines communicate with each other and with other immune cells to orchestrate a harmonious immune response. So these networks are incredibly dynamic, with cytokines acting in concert to fine-tune the immune reactions based on the specific context and requirements of the situation. So at the heart of these interactions lies the crosstalk between different cytokine signaling pathways. And cytokines can exert both direct and indirect effects on immune cells, influencing their behavior and function. For example, a cytokine can stimulate the production of another cytokine, which in turn amplifies or modulates the immune response. So basically, they can start a whole cascade of cytokine responses. So moreover, cytokines often exhibit something called pleiotropy and redundancy, meaning that one cytokine can have multiple effects on different cell types, and several cytokines can have overlapping functions. So this redundancy ensures robustness in the immune responses, allowing for flexibility and adaptability in the face of changing conditions. Also, cytokine networks operate within a complex microenvironment where various factors such as cell-cell interaction or tissue architecture and the presence of other signaling molecules influence cytokine activity and function. This microenvironmental context shapes the dynamics of cytokine signaling and ultimately determines the outcome of immune responses. Any kind of dysregulation or imbalance within cytokine networks can have profound implications for immune homeostasis and disease pathogenesis. Any aberrant signaling is implicated in a wide range of inflammatory as well as autoimmune disorders, and not to mention the progression of certain cancers. So their complex interactions form the basis of immune communication, ensuring that immune cells are able to effectively coordinate their efforts to protect the body from pathogens and maintain tissue integrity.
Great. So cytokines can be used for both therapeutic and diagnostic purposes. First, can you tell us how cytokine activity can be modulated for therapeutic purposes?
Yeah, absolutely. So you brought up a very interesting point that, cytokines can be used both as therapeutic as well as for diagnostic purposes. So cytokine activity can be modulated for therapeutic purposes by either enhancing or suppressing their effects. This can involve enhancing cytokine activity to boost immune responses against pathogens or even tumors, or blocking specific cytokines to alleviate inflammation in autoimmune diseases, or using targeted cytokine therapies to precisely regulate immune function.
You mentioned enhancing cytokine activity and blocking cytokine activity. Can you explain that a little bit more or give us some examples, please?
Sure, yeah. So some therapeutic approaches involve enhancing the activity of specific cytokines to increase the immune responses against pathogens or tumors. Like, for example, I was talking about interleukin 2 for T cell. So interleukin-2 can stimulate the proliferation and activation of T cells and natural killer cells, or NK cells, making it useful in cancer immunotherapy to boost anti-tumor immune responses. Another example can be the GCSF therapy, which is the granulocyte colony stimulating factor therapy. So GCSF promotes the production and mobilization of neutrophils from the bone marrow, making it valuable for treating neutropenia, which is essentially a condition characterized by low neutrophil counts. So that was activating or enhancing the activity of specific cytokines. So conversely, blocking the activity of specific cytokines can be beneficial in certain conditions like autoimmune diseases and inflammatory conditions where excessive cytokine production contributes to disease pathology. And these cases, they use inhibitors instead of activators. So tumor necrosis factor alpha, TNF alpha, we use inhibitors for those, using drugs like infliximab and adalimumab. They block the activity of TNF alpha, reducing inflammation and disease activity in conditions like rheumatoid arthritis, Crohn's disease, as well as psoriasis. Then another example is the IL-6 inhibitors. So inhibitors like tocilizumab and sarilumab, they suppress IL-6 signaling, providing relief from symptoms in diseases like rheumatoid arthritis and cytokine release syndrome, or CRS, associated with CAR T-cell therapy.
And you also mentioned targeted cytokine therapies. Could you also talk a little bit about that?
Yeah, sure. So with the advent of recombinant cytokines and cytokine analogs, researchers can specifically target desired pathways or even cell types. And these targeted therapies offer greater precision and reduced off-target effects compared to traditional cytokine-based therapies. And a lot of the times, these cytokines are used in combination with immunotherapy, and the cytokines can have a tremendous influence on immunotherapy outcomes.
How can cytokines influence immunotherapy outcomes?
So that's a great question. So cytokines influence immunotherapy outcomes mainly by enhancing anti-tumor immune responses, regulating immune checkpoints, inducing immunogenic cell death, modulating the TME or tumor microenvironment, and also contributing to treatment-linked toxicities such as the CRS that I mentioned before for IL-6 inhibitor. So basically, understanding these cytokine-mediated mechanisms is crucial for optimizing the immunotherapy strategies and also improving patient outcomes in cancer treatment.
Could you tell us a little bit more about that, please?
Yes. So enhancement of anti-tumor immune response, that's basically that cytokines can stimulate the activity of immune cells, such as T cells and NK cells. The example that I gave earlier was interleukin-2 therapy that has been used to boost the proliferation and activation of T cells, basically leading to improved anti-tumor responses in certain cancers. Then cytokines can also modulate the expression and function of immune checkpoint molecules, such as these are very common immune checkpoint molecules like PD-1 and CTLA-4, which regulate the immune responses and prevent autoimmunity, which is very important in cancer therapies. So immunotherapies targeting these checkpoints, like these immune checkpoint inhibitors, basically rely on cytokine-mediated mechanisms to unleash the anti-tumor responses and improve patient outcomes. Certain cytokines, such as tumor necrosis factor alpha, TNF alpha, and other interferons, can induce immunogenic cell death, ICD, which is basically a form of cell death that stimulates the immune responses against tumor cells. So immunotherapies that promote ICD, either through cytokine administration or other means, can enhance the efficacy of cancer treatments by stimulating the anti-tumor immunity. And then again, cytokines play a crucial role in shaping the tumor microenvironment, like all the other microenvironments in the body, and influencing factors such as inflammation, angiogenesis, and immune cell infiltration. So by altering the cytokine milieu within tumors, immunotherapies can modify the TME to favor the anti-tumor immune responses and pretty much stop or decrease the tumor growth and metastasis. And on the flip side, cytokine release syndrome, the CRS that I talked about, and other immune-related adverse events associated with immunotherapy can result from dysregulated cytokine production and cytokine activation. So managing cytokine-mediated toxicities is essential for optimizing immunotherapy outcomes and ensuring patient safety.
How can this knowledge be used to predict treatment responses and guide immunotherapy decisions?
So understanding the cytokine biology and its impact on immunotherapy outcomes helps in the identification of biomarkers for predicting the treatment responses, also guiding treatment selection and personalizing therapeutic approaches. So cytokine profiles can be used in treatment decision, monitoring therapeutic efficacy, and also facilitate the development of predictive models for optimizing immunotherapy strategies. And this knowledge basically enhances the effectiveness of immunotherapy and improves the patient outcomes in cancer treatment.
Could you explain it in a little bit more detail, please?
Yes, so I guess I basically threw out a bunch of words that may not mean a lot to the audience. So yeah, let me explain those things a little bit more. So cytokine profiles in the TME or peripheral blood can serve as biomarkers for predicting treatment responses and patient outcomes. And specific cytokine signatures may correlate with favorable or even unfavorable responses in some cases to immunotherapy, basically helping doctors identify patients who are most likely to benefit from a certain treatment. So that's why cytokine profiling can be crucial. And then I also talked a little bit about, I think, treatment selection. So the knowledge of cytokine-mediated mechanisms can inform treatment selection by guiding the choice of immunotherapy agents or even combination therapies. So that's basically the guiding factor there. And examples can be like, patients with tumors characterized by high levels of pro-inflammatory cytokines may respond better to immune checkpoint inhibitors, whereas those with immunosuppressive cytokine profiles may benefit from therapies targeting specific cytokines or cytokine receptors. So basically see the difference the cytokine profile makes in terms of treatment plans and guiding the treatment plans. And incorporating cytokine-related biomarkers allows for a more tailored treatment strategy based on individual patient characteristics. So by considering cytokine profiles alongside other clinical and molecular markers, doctors can optimize the treatment regimens to maximize efficacy and minimize toxicity. And monitoring changes in cytokine levels during treatment can provide real-time feedback on treatment. So I mentioned therapeutic monitoring before. So monitoring the changes in cytokine levels during treatment can provide real-time feedback on the treatment efficacy and help identify early signs of treatment resistance or disease progression. Serial cytokine measurements can guide the treatment adjustments, such as dose modifications or switching to an alternate therapy, basically to optimize the patient outcomes. And then there's predictive modeling. So machine learning models are now being used to predict how well immunotherapy will work for non-small cell lung cancer by analyzing the cytokines in a patient's blood. So these models look at the complex interactions between different cytokines to see how the body might respond to treatment. And scientists identify which cytokines are linked to overall survival and use them to create predictive models. So by training these models on both clinical as well as biological data, scientists have found that they can accurately predict how patients will respond to immunotherapy drugs like the immune checkpoint inhibitors, for example. And these models could help doctors choose the best treatment for each patient, reducing side effects and improving treatment outcomes, because that's mainly the goal here, and essentially a very, very personalized treatment. So for all of these predictions and profiling, the core of it all is standardized cytokine assays.
And why is it so important to standardize and validate cytokine diagnostic assays?
So standardizing and validating cytokine diagnostic assays is vital for ensuring accurate and reliable measurements across different laboratories and assays. This process basically instills confidence in diagnostic data, enabling informed clinical decisions, promoting research reproducibility, ensuring regulatory compliance, and maintaining quality control. This also facilitates the assessment of biomarker endpoints in clinical trials. So this is definitely where we come in as a biotech firm. With the quality and assurance on our cytokine products, therapeutic companies are using our products for assay standardization, drug development, drug targeting, as well as in the expansion of cells like T cells, NK cells, etc. So we offer RUO grade, research use only grade, as well as GMP grade cytokines to bridge the gap between research and clinical settings. And we manufacture our GMP grade cytokines in a GMP certified facility with strict GMP compliance. So that is important for the standardization of these diagnostic assays. So going back to your question, standardization ensures that the cytokine measurements are consistent and reproducible across different laboratories and assays. And validation verifies the accuracy and reliability of the assay results, reducing any kind of variability and ensuring confidence in the diagnostic data generated. So accurate cytokine measurements are essential for making any kind of informed clinical decisions regarding the patient diagnosis, prognosis, and treatment selection. So standardized assays provide doctors with reliable information to guide patient management effectively. And standardization and validation of cytokine diagnostic assays are also often required to meet regulatory standards for clinical testing. Adherence to the regulatory guidelines ensures that diagnostic assays meet the quality and performance criteria, enhancing patient safety and regulatory compliance. So that's basically the clinical setting. But in the research setting, standardized and validated cytokine assays are essential for reproducibility and comparability study findings. Let me talk a little bit about quality control as well. So standardized assays also incorporate quality control measures to monitor assay performance and detect any deviations or discrepancies. And these protocols are very important because they help identify and rectify issues promptly. You would really want to know if something is going okay or going wrong very, very quickly. That ensures the reliability and the validity of cytokine measurements over time. And the last thing that I mentioned was the clinical trials. Standardized cytokine assays are essential for assessing biomarker endpoints in clinical trials, evaluating novel therapies, including immunotherapies. Consistent measurement methods help with the data interpretation and comparison across multiple study sites, basically ensuring the validity and reliability of the trial results.
And finally, what do you think the future holds for cytokine diagnostics and their role in advancing precision medicine in immunotherapy?
So I think cytokine diagnostics have the potential to revolutionize precision medicine in immunotherapy by enabling personalized treatment approaches, then predicting treatment responses, monitoring therapeutic efficacy, informing combination therapies, guiding immunotherapy selection, and also driving advancements in clinical trial design. So as our understanding of cytokine biology continues to evolve and broaden, the technology continues to advance. And cytokine diagnostics will undoubtedly play an increasingly integral role in optimizing immunotherapy outcomes and improving patient care in the coming years.
Okay, great. So that's all of my questions. Thank you very much for coming on the podcast, Ritvika.
Absolutely. Thank you so much for having me here. It's always a pleasure.
It's always a pleasure to have you. If you'd like to find out more about the use of cytokines in immunotherapy, check out our In Focus with Cynobiological over on www.biotechniques.com. Thank you for listening and goodbye.